We have already considered the interplay between stimulatory and inhibitory G proteins in regulating the activity of adenylate cyclase. We now want to consider another GPCR signaling pathway. In this case, there are actually two secondary messengers produced from the same original molecule. This is the calcium and phosphatidylinositol 4,5-bisphosphate pathway. In other words, it involves the glycerophospholipid phosphatidylinositol bisphosphate, or PIP2, which you can see from the structure below is composed of two parts. It contains an inositol triphosphate, or IP3 moiety, as well as a diacylglycerol group. As in the previous example, the pathway initiates in response to hormone binding to receptor. The GPCR then binds and activates a G protein, as shown in the figure below, thereby causing it to exchange GDP for GTP. In this case, though, the G protein interacts with a different membrane-bound enzyme, phospholipase C. Once activated, this enzyme hydrolyzes PIP2, which is part of the lipid bilayer, as depicted below. Hydrolysis releases the soluble IP3 component into the cytosol, thereby leaving the DAG portion free to diffuse in the membrane. IP3 is the primary second messenger and diffuses through the cytosol until it reaches the endoplasmic reticulum. There, it stimulates calcium transporters to open and release calcium into the cytosol, as shown in the figure below. As the concentration of calcium increases, it binds to a soluble protein, calmodulin. The calcium calmodulin complex then binds and activates a protein kinase to initiate a kinase cascade. The hydrophobic DAG component diffuses through the plasma membrane and therefore also acts as a second messenger. It activates another membrane-bound enzyme, protein kinase C, or PKC. This kinase phosphorylates channel proteins that control the flow of calcium in and out of the cell. The enzyme is named for its function, protein kinase C, C for calcium. By controlling the flow of calcium ions, this second messenger system can produce sustained responses even when the supply of calcium in the intracellular reservoirs becomes exhausted.